Thank you. Thank you everyone to having me to share our work on the multi-omic integration analysis of cell type specific regulatory networks in insulin resistance in different ancestry populations. A little bit of background of the analysis. Type 2 diabetes is one of the very common chronic diseases impacting over 30 million of Americans. And in type 2 diabetes, patient cells stop responding to insulin and present insulin resistance. That is, the loss of insulin sensitivity due to the impaired regulation of the blood sugar metabolism. However, so far there's no cure for this disease, and our knowledge of the molecular mechanisms underlying insulin resistance is still very limited. So for our analysis, we are motivated by a number of scientific questions, such as what are the landscape of cell type specific gene features and gene coexpression network and regulatory networks in response to insulin resistance in the adipose and muscle tissues, which are the major glucose responsive tissues. And how are gene networks preserved in different human populations? Because we know certain ethnic groups have higher susceptibility to type 2 diabetes. And then how are the gene network modules regulated by genetic variants, such as the expression quantitative loci, which I will describe later. So to answer such questions, we perform an integrative network analysis by using tissue samples from 3 cohorts. So we collected adipose tissue and muscle tissues from one African American sample with 256 subjects, and one European population with over 700 subjects, and lastly, a smaller European American population with about just below 100 individuals. So from these adipose and muscle tissues, we perform gene expression and profiling to evaluate expression changes of the gene. And for each subject, we also collected the insulin sensitivity related phenotypes. And also we profile the DNA's genotyping of each individual. So with this data, we performed 2 types of network analysis. The first one is the coexpression network. From this network analysis, we identify the co-regulating gene network modules, and then we associate the network modules with insulin sensitivity related clinical phenotypes to see what gene pathways relating to insulin sensitivity. And then we also characterize the cell types of their gene modules by a single cell RNA sequencing data. Also, we compare the gene modules between ethnic groups and between tissues. In the second analysis, we utilize the Bayesian network to identify the regulatory relations among the network modules. From the network, we find the network key driver genes that are implicating in the response to insulin sensitivity or insulin resistance. And lastly, we perform experimental validation on those predicted key drivers. Before going to the network analysis, I'll just briefly describe some simple analysis of the correlation between the gene and insulin sensitivity related phenotypes. So in the adipose tissue, we found several thousand genes either positively or negatively correlated with the insulin sensitivity related measures. Here we use 2 types of insulin sensitivity related measures. One is SI, the other is Masuda. They are both glucose tolerance tests, have different complexity. And then we also correlate genes with the BMI body mass index. For the muscle tissue, we identify several hundred genes associated with the insulin sensitivity and BMI. Even though there are the number of genes in the 2 tissues are quite different, we found a significant overlap between the 2 tissues. For example, as you can see that the positive associated genes from adipose tissue for Masuda is significantly overlapping with the positive associated genes in the log Masuda from muscle. And when we look at the gene functions of those insulin sensitivity related genes, what we call ISG, You can see they are falling into different categories. Specifically, we found mitochondrial translation and the ribosome genes are negatively associated or downregulated in the patient. However, the genes involving in immune response, like a vacuole, are activated or upregulated in with insulin sensitive, insulin resistance. And so from this analysis, we know that insulin sensitivity associated genes regulated various functional groups. So we further used the multi-scale gene co-expression network to organize those genes into pathways. Here we use a tool called Magina, which is developed by my colleagues at Montana to identify the co-expression, co-regulated gene modules. Here, this figure in the middle is a sample network. Each dot is a gene, and the gene is colored by the module membership. And then with the modules, we better characterize their functional pathway, enrichment for ISGs, and trait correlation or cell type specificity by comparing with single cell data. And also, we perform module preservation across tissues and ethnicities. So here, I summarize some of the results from the adipose tissue network in the African American ancestry populations. This samples plot just shows the hierarchy structure of the coexpression network modules. So here, the module is a gene cluster, as I didn't mention clearly. And then the modules are colored by the association with the Masuda index. And then highlight these three groups of hierarchy modules. So on the first group involved module M9 is relating to the response to hormone. And the second group of modules involving module M6 is relating to immune response. And the third group involving module M5 is relating to mitochondrial genes. And since we know genes expressing different cell types, utilizes single cell RNA sequencing data from adipose tissue to check the cell type specificity of the modules. And we found many of the modules specific to certain cell types, such as adipocytes and those are stem cell related progenitor cells and B cell, endocyte, dendritic cells. And specifically for the module M5 is adipocyte related cell type, and module M9 is also enriched in adipocytes. And for the module N6 is relating to a number of immune cell types like macrophage, monocytes, neutrophil, and K cells. Similarly, we analyzed the network in European populations. Here I highlight two groups of network modules. The first group involves module M15, which is relating to immune system processing. And the second group involving module M12 is relating to mitochondria. And again, we check the cell type specificity of the modules. So when we compare African American population networks and European population networks, we found many top pathways are common, such as mitochondria and the immune response. So let's say there's a lot of preservation. So between the two populations. So formally test the preservation of the network modules. We first utilize preservation test by looking at whether the genes in a module from a particular population remain highly or significantly intact in another population. So as you can see, those modules with very high preservation scores tend to strongly associate with the normal Masuda, either positively or negatively. And such as the module M5 and N6, I just mentioned previously. And then we also just directly intersect the modules between the two populations to see whether they have a lot of common genes in the same between modules. Recently, for the module N6 and 30 from the African American population, highly overlapping with a number of modules from the European population network, such as the N15. And then when we look at the networks between these highly overlapping modules, M6 from the African American adipose network, and M15 from European adipose network. There are a lot of common genes, as you can see from this orange link. And also we checked the network modules on African American samples. When we look at those how the modules associated with the Masuda, we found the mitochondrial pathways are highlighted, but we don't see much immune response related to gene pathways. So I would suggest they are like a common network between tissue types, but they are also tissue specific. In summary, of my first part, so we conduct a systematic cross-population and cross-tissue analysis of the cell type specific regulation of gene coexpression networks underlying insulin resistance. And many top-ranked insulin sensitivity associated modules were preserved across different ethnic populations. And those insulin sensitivity associated modules highlight immune response, mitochondrial and hormone response pathways in adipose tissue. And we found tissue-specific network response when comparing adipose and muscle. From those, we can get an landscape of co-regulation of the gene. However, we don't know the regulated relationship among the genes. So to tackle this issue, we turn to the Bayesian network analysis. The goal here is to identify the true hidden direct relationship among a network of genes, such as here the gene ABC. There is a relationship from A to B and B to C. But in the biological data, what we typically observe is bidirectional correlations, and they are present many micro-equivalent population structure that cannot simply be distinguished based on the gene expression data alone. And many of the network structures are distinguishable, but have quite a contradictory relationship. We need some prior knowledge to break the micro-equivalence. Unfortunately, in the biologic data, particularly in the gene swing data, we have a natural source of a genetic prior. That is, the DNA variation exists in our human DNA. Here, our DNA. Among the human DNA chromosomes, over 99% identical. Each individual have many differences in their DNA, and most of these differences are just single base substitutions, such as in this figure. We call those single nuclear polymorphisms. And those genetic variations are inheritable and are the root cause differentiating individual characteristics and phenotypes, such as the disease susceptibility. And many of those genetic variants are having no functional consequence. However, those SNPs located in the genes or in the regulatory region of the genes have a significant functional impact, such as regulating the gene expression. So when we associate the gene expression with the SNP genotype, we can find whether a particular SNP is potentially regulating the expression of one or more genes. Here, the figure to the right, just an example of such a SNP. And the genes, the expression of the gene ECHDC3 is reduced in the CT and TT genotype of this particular SNP compared to the CC genotype. So there's such gene expression associated SNPs are called expression quantitative trait loci. And depending on the location of the expression quantitative trait, they can be classified into cis-eQTLs if they are located close to the gene, or trans-eQTLs if they are far away. And since the genetic variation are naturally occurring in the population, they represent the natural randomized perturbation that can be used to inform causal relationship among the genetic phenotypes, such as the gene expression traits. For example, we can assume that if the gene is containing a cis-eQTL, then this gene is very likely to be self-regulated. However, a gene has a cis-eQTL is likely to be regulated by another gene. And then for the genes associated with the same transcriptomic, we can perform a formal causal or inference test to infer the causal directions. For this, we utilize a tool called causal inference test, or CIT. With this tool, we can compute the probability of different models involving eQTL loci, like a locus L, and two genes A and B also with locus. There are a number of models we can evaluate, including two causal models and one independent model. And then another type of genetic prior we can utilize in the network analysis is the transcription factors. Gene expression is the process of transcribing DNA genetic information into mRNA, and the mRNA is transcribed into protein to perform the functions. And the transcription factors are the regulated proteins that help turn the specific gene on or off by binding to the specific sequence of DNA nearby the targeting genes. So the transcription factors are actually controlling the expression of the genes in different cell types under different conditions. There are many databases providing rich information about transcription factors and their targets. So with the advice from eQTLs and vector and type relationships, we utilize computational software LimbNet to build Bayesian causal networks. This tool, LimbNet, was developed by Professor Jim Zhu and This tool starts with the random network structure of all of the genes in the data, typically over 10,000 genes or 20,000 genes. And then it iteratively updates the network by multi-color simulation and grid search to avoid local optimals. So this tool will work thousands of networks, each started with different random structure, and finally compute a consensus network out of all of the network. Once we derive the Bayesian network, we can project the machine coexpression network modules and ISTs we described previously onto the Bayesian network to identify if those modules or ISTs are enriched in the network level or particular network hub. So we will call such hubs as key drivers. Yes, so here's some results from the expression network analysis. For example, this gene ECH, EC1, so DC3, previously mentioned, is positively associated with the Masura index, and then it's cis-regulated by a SNP. And then when we look at the network modules and the key drivers, we found some of the network modules from adipose tissue enriched for expression QTL genes, such as M5 and 9 we mentioned before, and also like 7 of the network key driver genes from those resistant related modules are showing enrichment for eQTL genes in the network level. The figure C is just an example network of finding the leptin encoding gene LP. So here the orange nodes are the eQTL genes. So the enrichment of eQTL genes in the network modules and the key drivers suggesting they are likely involved in the genetic regulation of the gene network. And then to test if the network key drivers we identified indeed performing regulatory roles in a tissue-specific manner. So we selected 5 key driver genes from our analysis, including 3 genes from the adipose side specific module M5, that is ACADM, ALDH6A1, and HADH, and 2 genes from the immune response module M6, that is APTM5 and PAL. We first check the cell type specificity of those genes. So when we check the expression of those genes in the adipose tissues, for those 3 genes from the adipose side specific module, their expression is much higher in the adipose side fraction compared to the vesicular fraction. So adipose side fraction is colored by the orange, and the vesicular fraction of the adipose side, so adipose is colored by blue. On the On the other hand, key drivers PM5 and PEL are more enriched in the vascular fraction of adipose. The vascular fraction of adipose include a cell type like pre-adipocytes, vascular cells, and the immune cells. Since two genes are from immune resource modules, so this enrichment in the vascular side, vascular fraction of adipose make sense. And then we also we also confirm that ACADM, LDH61, HADH expression increase throughout the differentiation of mature adipocytes. And the PTM5 and NPL expression level increase throughout the differentiation of P1 macrophages. So out of these key driver genes, NPL is a novel key driver, and its function in obesity, glucose metabolism has not been studied. To further assess this gene, we developed a knockdown system to suppress the expression of NPL, THP1 cells, and also in PMA-induced macrophage cells. THP1 cell is more like a monocyte cell. Most monocyte macrophages are immune cells. We can see that by using shRNA or targeting NPL, the gene expression is significantly reduced in both the THP1 cells and also the PMA-induced macrophage cells, suggesting that the knockdown system works. Then we utilize the RNA sequencing to characterize the genomic response induced by knocking down NPL in macrophage cells. And from RNA data analysis, we identified over 1,000 differentially expressed genes, and we confirm the expression of NPL is significantly reduced and knocking down. And the genes downregulated by the NPL knocking down is highly enriched for cytokine production related genes. These are the cytokines type of immune vectors. And then the upregulated genes are involved in the extracellular structure and organization. And then we check whether those genes from NPL knockdown enriched in the network neighborhood of the NPL genes. And indeed, we found a significant enrichment of the disease in the network neighborhood of NPL genes, as shown in this figure, which illustrates two-layer neighborhood for NPL. And many genes are either up or down regulated, as represented by red and blue color in response to the NPL knocking down. So this is suggesting that we knock down signature from our validation experiment confirmed the network prediction by our integrative network analysis. Okay, in summary, we constructed Bayesian networks by incorporating expressing QTLs, key transcription factors, and gene expression data. By integrating Bayesian and networks, we identify driver genes for cell type specific insulin sensitivity associated modules. And we also experiment to validate the cell type preferential expression of selected network key drivers. Features from knocking down one key driver gene in peer resemble the prediction by our integrative network analysis. So the analysis presented here has been published recently in the journal Cell System. Lastly, I want to thank my colleagues, Professor Bin Zhang and Chen Xu. Bin Zhang is my mentor. And this work was carried out in collaboration with Dr. Swapan as from Wake Forest University School of Medicine. And this study was also supported by enriched grants. So thank you for your attention and welcome your questions.